In today's class, we are going to study about the bacteria, which is the only member of kingdom Monera. This kingdom has the maximum nutritional diversity. First, let us see how we are going to draw the bacterial structure. So let us study in detail about the structure of bacteria. The outermost layer, which is in brown color, which I have drawn here, is nothing but glycocalyx, which is the attachment. The main function for the glycocalyx is attachment, or we can say also for protection. And this glycocalyx can be of two types. In case of bacteria, it can be smooth, that is first one is smooth, that will be looking like slime layer, and in some cases it is hard. Outermost glycocalyx will be hard, which looks like a capsule like structure. So the main outer part is, which is the brown color here, it is a glycocalyx which I have surrounded with the brown layer, which is glycocalyx. The main function is attachment or protection. Okay, next is the comes here is the second layer, we can say is the cell wall, which is the outermost layer in case of bacteria and is made of peptidoglycan. Peptidoglycan. And the third one inside here, you can see it is plasma membrane. Plasma membrane, which is made up of lipid bilayer. Inside the plasma membrane, you can see the matrix which is made is called as cytoplasm. Okay, then you can find here and there some ribosomes. You can see here or here, and these are embedded inside the cytoplasm, which are called as ribosomes. And especially in case of the bacteria, we can find 70S ribosomes, which help in protein synthesis. Now let us come to the main nucleoid which is present in case of bacteria, that is nothing but this nucleoid. This is the nucleoid, which is nothing but the DNA, double stranded DNA, which is circular, nothing but dsDNA, that is double stranded DNA. Or we can call other name for the nucleoid which is present in case of bacteria is genophore, a 4 carrying a gene, or we can also call it as prochromosome. So if a question is asked what is a genophore, we can even simply you can say that it's a bacterial DNA. Genophore is nothing but the bacterial DNA, or prochromosome is also nothing but the bacterial DNA, which is double stranded DNA. Okay, or one more name given to this one is incipient nucleus. Incipient nucleus. Now in case of this circular DNA, there are no histones present in case of bacterial DNA, no histone proteins. These histone proteins are required, that is, these are the proteins which are required for packaging this DNA. But in case of this bacterial DNA, there are no histone proteins present. I have mentioned here is ribosomes. Now you can see one more here. There are some ribosomes which are single in case of cytoplasm, and there are roaming in cytoplasm, but there are certain ribosomes which are attached on a single strand. You can see here. You can see here. Now this one I call it as polyribosomes. Why we are calling it as polyribosomes? Poly means many. I have mentioned only two here. It can be more than two also. Poly means many, and ribosomes. Polyribosomes. Ribosomes which are attached on a single mRNA strand. This is the single mRNA strand. That is, okay, we are calling this as polyribosomes, or we can give the other name as polysomes. Simply we can call them as polysomes. Next, I have given an attachment which is coming inside, protruding inside here, attached with the plasma membrane. We call them as, this one is called as chromatophore. Chromatophore. Now you can, by the name itself, you can easily understand that this chromatophore, chroma means green in color, phore, a phore which is green in color and which performs the process of photosynthesis. Chromatophore is nothing but this pore will help in performing the process of photosynthesis. We have one more protrusion which is coming or bending inside with the help of this plasma membrane, that is plasma membrane which is bending inside here. We call this part as mesosome. This is called as mesosome, and it has many functions. Let us see one by one what are these functions. First one is it helps in the cell wall formation. It helps in cell wall formation. Okay, and also this mesosome will help in reproduction during the reproduction time, and one more is respiration. The other function is respiration. The mesosome also helps in respiration. Then next is replication of DNA. During the process of replication, this mesosome will help in the process of replication of DNA, and also for secretion of the particles, and it also increases, okay, increasing the surface area, as well as the last one but not the least is it is having an enzyme activity. These are the main functions of carried out by mesosome. That is, mesosome is the one structure which is bending inside or internally, we can say, from the plasma membrane, which the main functions are cell wall formation, reproduction, respiration, replication of DNA, secretion, increasing the surface area, and as well as it has the enzymatic activity. Right, now let us move on to the next one. Here we have one more. This is the flagella. This is the flagella. Now here you can find the flagella. I haven't drawn the flagella as simple as here. This is the complicated structure we can find here. If you can see here, I'll draw one more part here, the flagella. Like you can see, the flagella is a hook-like structure. You can see here, it's looking like a hook which is having a filamentous structure attached to it, and then down you can find the basal body. Like it looks like this, as if some circular parts are attached to the stalk-like structure. This is what is called as the basal body. This part is called, this part is called as basal body, and this part of the flagella is called as hook, and here the flagella, which is motile, it is called as a filament. Together this is the main structure of flagella which I have shown here, on which is attached to the bacterial cell wall. Okay, this was about the flagella. Moving on to the structures which you can find apart from the flagella, you can see the protrusions outside which is attached with the glycocalyx. These are nothing but pilus, called as pilus, or we can say that as fimbriae. Now these pilus or fimbriae are very long in structure and or less in number. The next part here you can see here is these are the gas vacuoles. So how should I show you here? Right. Okay. These are the gas vacuoles. Now which help in buoyancy, or we can say rotation of the bacteria. Next coming here is the next part is the one apart from the bacterial DNA. In the bacteria we can also find plasmid, that is an extra circular double stranded DNA, we can say. This is how I should mention here. Okay, let me show here. This is what we are calling it as plasmid, which is circular in shape, or we can say extra chromosomal DNA or extra circular double stranded DNA, which can be sometimes present and sometimes may not be present. So it is circular and double stranded DNA. So this was about the structure of bacteria. I hope you understood the class. Thank you.